Happy Thursday, guys. I hope things are going well so far today. I decided to put together a little video to be able to give us the opportunity to go over our quiz, hit some major gems of wisdom, then talk a little bit about our homework because that's going to be essential in really being successful with our lab today, and then actually be able to go over the components of your lab so that we can be most successful. So first thing we want to do is, of course, turn to our Gems of Wisdom page, and we're going to address some of the major things that seem to show up on our quiz. So the first item from our gems that I wanted to talk a little bit is about the structure of the cell membrane. So the primary molecule which makes up the cell membrane is going to be our phospholipids. So our phospholipids are going to be the structures which contain the phosphate head, which loves water. They are hydrophilic. They love water, so they're always going to be where water is. There's going to be water inside the cell, and of course there's water outside the cell. And then on the inside we've got our lipid tails, and those lipid tails are going to be hydrophobic. So phobia means that we are scared of, and so the lipid tails, they don't mix well with water. So it's really important to understand that the main molecules that make up the membrane are phospholipids, and they contain the phosphate heads and the lipid tails, and because of their difference in chemistry, that's why the tails are going to find their way on the inside. Now, a second molecule which is also important to the membrane is going to be our proteins, those transmembrane proteins which are going to be responsible for transporting things across that membrane. Okay? Now, the next item has to do with our diagram of our cell placed in some salt water. Now, in this particular case, we're given the concentration of solute. The solution has 10% salt and our cell has 1% salt. So in order to see what's going to happen to that cell, we have to know how much solvent, how much water is present. So if I have 1% salt, that means I'm going to have 99% water. If I have 10% salt, that means I have 90% water. So where is there more water? There's more water in the cell, so water is going to go from an area of high concentration to an area of low concentration, and so that water is going to leave the cell and it's going to shrink. One way that we can try to remember that is when you have a really salty solution, cells tend to shrink. Kind of a good turn of phrase is that salt sucks. It's going to suck the water out of the cell. Now, when you have a solution which has a high concentration of solute, in this case 10% versus 1 in the cell, this solution is referred to as hypertonic because it has lots of dissolved solute. So our next gem of wisdom is going to come from our fill in the blanks. It's going to be the second fill in the blank, which asks, what is the word or the term that's used to describe when a cell takes in a large amount of things? Now, some of you said that it was bulk transport, and I said that was fine. Some of you said that it was a form of active transport, and I gave kind of half credit for that because, yes, it is active transport, but we're looking for something a little more specific. When something goes into the cell, that's going to be endocytosis. So like this amoeba is forming its pseudopods and bringing in a chunk of food. We call that endocytosis. And then, of course, if our amoeba decides that it needs to get rid of a big chunk of waste and it's going to push that waste out of the cell, that's going to be exocytosis. So those are some words that you'll definitely want to add. And of course, to remember which is which. Endo means going inside. Cyto means cell, inside the cell. And exo, that means we're going to leave the cell. Lastly but not leastly, I wanted to talk a little bit about our short answer. So in this particular case, we were giving a little kind of square cell, and we had some molecules represented by these little O's. And you were first asked to draw arrows to show which way those little molecules might diffuse. Remember, diffusion is the movement of molecules from high to low concentration. So you should have drawn the arrows of our molecules leaving that cell. Then you had to explain what was going on. It was important that you talked about the fact that inside the cell there was a higher concentration and it needed to move to where it was a lower concentration in order to reach equilibrium. So you needed to be able to address the concentration difference that was driving that process. So these were just the 4 major gems that we were interested in. I hope these were helpful. If you need to, you can pause the video so you can finish writing these gems before moving on to checking our homework. Alright guys, now it's time for us to take a look at our homework from last evening, where you were asked to review the stages of mitosis and make sure that you're able to identify the nuances between each of those stages and kind of talk a little bit about what was going on in those stages. So the first stage that we have to identify is interphase. And remember, interphase, when scientists first examined those cells, they thought it was resting because they couldn't see any major things happening in the cells. So interphase cells look kind of normal. They look like a regular cell with an intact nucleus, like cell 4. But remember, there's a lot of stuff going on. It's certainly not resting. The cell is growing, eventually the DNA is copying, and then we're going to prepare for mitosis. So then we'll have cell 5, cell 6, cell 8, cell 10, cell 13, 15, 16, and 18. So there's an awful lot of cells undergoing interphase. So after interphase, the cell is going to begin mitosis. And the first stage of mitosis is prophase. Lots of stuff happens in prophase. One of the major things that has to happen is the spaghetti-like chromatin needs to be packaged up into the rod-like chromosomes, which we see happening here in cell 14. Spindle fibers will form, and in order for our chromosomes to move about, we need to dissolve the nucleus, which is happening here in cell 2. Next up, after we have dissolved the nucleus and the spindle fibers are formed, it's time for the chromosomes to be pushed to the middle during metaphase. And so we see that happening here in number 1. We see that happening here in number 11. And then we see that happening here in number 17. Now once they're at the middle, we're ready to then divide our cells evenly. And so the sister chromatids are going to split apart equally, half going to one pole and half going to the other, to make sure that all the new cells are going to have equal amounts of chromosomes. And we see that happening here in 7 and 9. Lastly, but not leastly, once we get to the poles, it's going to be important then for that genetic material to be then enclosed in a new nucleus, and our chromosomes then will unpack back into chromatin. And then finally, cytoplasm will split through cytokinesis, and of course these are plant cells. We're going to be forming a cell plate. And so we see that occurring both in 3 and 12. So one of the things that's kind of interesting when we examine the stages of mitosis, really prophase, here like 14, is almost like the opposite of number 12, telophase. Prophase, we have our nucleus eventually dissolving, which is kind of interesting. So our nucleus is dissolving, and then in telophase, the nucleus comes back. In prophase, our chromosomes are packed as into the rods, but in telophase, they go back to being threads. They're like completely opposite of each other. So in just a minute, you guys will then be transitioning into a rather large lab where you're going to be able to see real cells in these stages, and then determining how long it takes for the process of mitosis. So you may pause the video now as you guys kind of transition and pull out those lab sheets, and when you're ready for instructions, we can hit play again. Okay, so now it's time for us to get at this lab and see if we can actually observe mitosis, the actual cells undergoing that process of division. To do that, we're going to use these microslide viewers and a slide set. So each pair should pick up a viewer and then a little folder, an information folder which contains a set of slides. On the end of the slide, there's an arrow that points to the direction that it needs to enter into the microslide viewer. Just like on a microscope, there are actually some focus knobs, which is going to allow you then to focus on what you're looking at. So when you put this microslide in, you're going to see a photograph of cells in different stages of mitosis. Oftentimes, they're actually going to be labeled. So what we're actually looking at here is a cell F. Okay? What you're going to need to be able to do then is to draw that cell. You want to make sure that you draw the actual cell wall and then the chromosomes in whatever stage they're in. So you'll have your cell wall and then whatever is happening to the nucleus. Don't just draw the chromosomes. Now, each of the slide sets has a number. So this I think might be number 6. It will correspond with the folder that you received when you picked up your slide set. That folder then will help you because it will explain what's going on in that particular moment. Now, there are a lot more pictures on our slide set than there are actual, main stages of cell division and mitosis. And that's because in some cases they give you an earlier picture or maybe a later picture of that particular stage so you can see the transitions that are happening in that cell. If I were you, I would focus on not the early ones, but if there's one which talks about it being the late version, that's probably going to give you a more dynamic image of that particular stage. There are questions associated with each picture. You can use your notes to answer those questions, or of course the folder has a little blurb on that particular stage that also might be of assistance. When you finish with part A, you'll be ready to move on to part B. In part B, you're going to be trying to figure out how long it takes for cell division and mitosis. You're going to figure out kind of the time it takes for a cell to go through complete division. Our cells of choice are going to be onion root tips. Onion root tips are rapidly growing cell types, so we're going to be able to see lots of cells undergoing division. In order to do this, you're going to obtain a photograph of those onion root tips from your substitute and an expo marker. The colored copies of these slides are actually laminated, so we're going to be able to use our expo marker to identify the cells and their various stages. So we're going to need to be able to identify the cells in their stage and count up how many there are in each stage. If we're looking for interphase, we're going to be looking for cells which have an intact nucleus. Looks like a regular cell, and so those will be the ones we'll count as interphase. For prophase, we're going to be looking for cells whose nucleus seems to be breaking down, and we can see rods instead of maybe like threads for our genetic material. So that will be our prophase cells. Our metaphase cells will be those which are having our rod-like chromosomes going to the middle of the equatorial plane, so they're going to be lined up at the middle. In anaphase, the sisters have broken apart, and so we're going to be looking for cells that are broken apart. Telophase, we might be looking for like 2 brand new nucleuses that are formed, like little tiny cells with brand new nucleuses, or maybe even a weak cell plate being built in between for our telophase. Now, once we've identified all the cells that we can, because if there's any of them that are fuzzy and we don't know what they are, we'll just ignore them. So all the ones that we can identify, we will, and we'll record the number that we count over here on our data table. So let's just say, for instance, I counted 70 interphases, maybe 30 prophases, 10 metaphases, 5 anaphases, and 3 telophases. So how many total cells did I count? 70 plus 30 is 100, 110, 115. I counted 118 cells. So we're going to assume that the number of cells in that particular stage is equal to the fraction of the time that it's in cell division. For onion root tips, it takes approximately 720 minutes for it to complete. So to figure out how long it's in each stage, what you're going to do is you're going to take the number in that stage. So like for interphase, it would be 70. You're going to divide that by the total number, which would be 118, and then multiply by 720. So it's the number of cells in the stage divided by the total number of cells, and then we'll multiply that by 720, and that's going to give us the time in each stage. From that, you'll be able to create a pie chart that kind of illustrates the differences in time, and then compare your data of a plant cell to a salamander kidney cell, which is an animal, to see if you see any patterns. And then, in preparation for Friday, you'll be able to compare normal chicken cells to cancerous chicken cells to see if there's any other patterns with regards to time in each stage and mitosis. I hope things go well. You'll be finishing this up for homework. Tomorrow, Mr. Parks will be your substitute. And if you have any questions, I'm sure he'll be happy to answer them. Good luck and have a great day.